Okay, hi everyone. I think we're going to make a start. So welcome to this second webinar in our new OBON series, which is introducing new and existing OBON endorsed projects within the UN Decade of Ocean Science for Sustainable Development. So I'm just going to start with a brief overview presentation on OBON. By the way, I'm Sophie Sieve. I'm the CEO of POGO, which is the Partnership for Observation of the Global Ocean, which is the lead organization for OBON. Sorry, I'm just trying to find the right window to move through the slides. Okay, so you can download our new OBON strategy at the link that's shown here with the QR code as well to find out more about OBON. But in a nutshell, our vision is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. And OBON's mission is to create a hub for biomolecular measurement of marine life. So we have four main objectives, which are all quite ambitious. OBON is set to be at least a decade-long program, and very probably will extend beyond that given the ambitions that we have. So number one is to build a coastal to open ocean multi-omics biodiversity observing system, which will involve developing autonomous sampling and analysis capability and promoting methods and technique development. We aim to enhance marine omics data systems and ecosystem models by adding biomolecular components. We want to partner with marine conservation and management specialists, policymakers, the private sector, and ultimately to address sustainable development. And a very important part of our objectives and mission is also to develop and transfer capacity through training programs and also funded equipment programs that will be supported by development and aid agencies as well as philanthropy. This diagram shows our kind of overall strategy, which is articulated kind of around those four objectives. With the sort of focus areas of developing networks and resources, innovating through technology development, enhancing information use through models and digital twins, as well as data management. And our ultimate goal, which is very much in line with the UN Decade, is to inform policy and ultimately a sustainable future. So the way the Ocean Decade works is through endorsement of projects that sit underneath broader kind of umbrella programs. And OBON has been inviting proposals for endorsement since the beginning of the decade in 2021. So we now have 20 endorsed projects that can sit underneath OBON. And it's these projects that we're showcasing in this webinar series, and we've tried to group them by themes so that each webinar focuses on a particular area of OBON's strategy. So this map shows our coverage in terms of sampling locations where biomolecular observations are being carried out by our projects. So we're getting fairly global with our coverage, but still fairly restricted to coastal areas and also with gaps in certain parts of the world. In terms of our structure and organization, we have a coordination office, which is currently supported mainly by the POGO Secretariat. And we have Luz Rodriguez, who is currently working as our program manager. We have a scientific advisory committee and a sort of subcommittee, which is the executive committee, which support OBON's objectives and the carrying out of our strategy. We're in the process of setting up a user advisory committee, and we have a number of working groups that are in various stages of development, some that have been going for quite a while, like the technology development and capacity development working groups, one on protocols, which is actually transitioning from the BeBOP project that Katie will be presenting today, and we plan to have a policy working group very soon. We've been running a number of workshops, and we have others being planned. So we had a very interesting workshop at the Westpac Symposium in Bangkok earlier this year, which has really helped OBON to reach out to a new part of the community in sort of around the Western Pacific and Asia, which we previously didn't have so much involved in OBON, so that's been really beneficial. We're planning a workshop on eDNA for fisheries management for next year, and also a capacity development workshop, also aiming to establish an eDNA network in Latin America that's planned for Colombia in tentatively in 2025. So that was just a very quick overview, and here are some ways that you can follow us and contact us directly. Margaret Leinen is the chair of the OBON Scientific Advisory Committee. She's also in this webinar, and we're both happy to communicate with anyone who's interested in OBON. Okay, so for today, we'll have 3 OBON projects being presented. So we'll start with a pre-recorded presentation by Miwa Takahashi from CSIRO, presenting the Making eDNA Fair project. Miwa is actually here, but provided a pre-recorded presentation in case there were any issues. So she'll hopefully be around to take questions as well. And then we'll have presentations by Anaïs Lacoursière from the Department of Fisheries and Oceans Canada on the project called Guidance on Optimal Timing for eDNA. And then we'll finish off with a presentation by Katie Pitz from Mbari on the BeBOP project, which stands for Better Biomolecular Ocean Practices. I think one of the best acronyms we have in OBON at the moment. So I will now hand over to Luz, who will share Miwa's presentation. And just to say that there'll be time, hopefully, for questions in between all the presentations. So if you have burning questions for any particular speaker, we'll be taking those after each presentation, and then hopefully have a bit of general discussion time at the end.
Hi everyone, my name is Miwa. Thank you very much for having me today for this seminar. I would like to talk about the project to make eDNA data fair. FAIR stands for Findable, Accessible, Interoperable, and Reusable. So first of all, why do we need to make eDNA fair? It's because of the number of environmental stresses around the world. We need tools to support better level of biodiversity monitoring, and eDNA is a very powerful tool for that. And because of the robustness of the eDNA tools, a number of eDNA studies have been increasing very rapidly, as you can see in this graph on the right hand side. And this is just on aquatic eDNA studies, so water samples on a macroorganism. So if we include also other type of samples, for example, sediment, soils, and air, on microorganisms as well, the number of studies per year published could be well over 1,000. And each of these studies produces tremendous amount of eDNA sequences and biodiversity species occurrence data. And those are very, very important and valuable for biodiversity monitoring and conservation. If we have all this data in a fair manner, what can we do with it? We can do all sorts of things. For example, for the studies at extended time and space, that is impossible to do within a single project. We can also increase species identification accuracy by reanalyzing eDNA sequences using updated reference databases. We can also enhance collaboration through by reusing the data from different projects, different organizations, different countries. And through these activities, we can generate knowledge at speed, and ultimately we can support decision making for conservation. So, and where do those valuable data go once studies are completed? Unfortunately, many of them are single use only, getting lost somewhere in this data map. But some data are made available in various platforms like this, in various data formats. And because the formats are different between the databases or between the studies, those data are still not fair. We cannot collect them together and reuse them. Another reason why the data is still not fair is because data are shared but half empty, as you can see in this the title of this paper. And for example, some researchers might share eDNA sequence data in supplementary materials of journal papers or Dryad, but it might be missing the sample metadata, and in that case, data is still not reusable. So these are the challenges that we are trying to overcome in our project. Here is the infographic of our project. So eDNA process, starting from the sampling and then data generation, species identification, and currently the data is not standardized, so there are those pieces of puzzles from different studies cannot fit together. And we are working in this middle part to change this data life cycle. The outcome is the pieces of puzzles can fit together, so we have one big eDNA data set, and then we can also plug in with other type of biodiversity data. So I'm going to walk through this middle part, what we've been doing in the last 1 year for this project. So first of all, collaboration is essential to achieve this goal. We have now this FAIR eDNA working group. We are a group of over 25 people, a lot of eDNA researchers from multiple labs, and also database managers, because cyber infrastructure here is one of the key elements to make eDNA fair. And we also have journal editors and publishers on board because we want to implement the FAIR eDNA guidelines into journal publication policy. And this is going to be a very effective way to influence scientists' practice towards FAIR data principles, because most eDNA data goes through the gateway of journal publications. So we're currently working with Wiley Publishers, which has environmental DNA, molecular ecology, ecology and evolution, so very relevant journals for eDNA studies. And they're really keen to implement the FAIR guidelines into their publication policy because they see the value of FAIR data and eDNA data. Now moving on to this data standard format. And so with the working group members, we're now establishing a data format and a vocabulary. And the key focus here are the complete information and machine readability in order to achieve this I&R, the interoperability and reusability. The first thing we did was to define what types of data are required. Here you can see the several different types of data, and one is, for example, study metadata that contains information about whole project, sample metadata that contains information of each sample, and there are multiple tables, OTU tables, species assignment information, and so on. And once we defined what the required data types, we developed the eDNA metadata checklist, which is basically the list of information required in the study and the sample metadata. This is the small fraction of the checklist, and here you can see the section. It starts with the project information, and here you can see the sample collection methods, storage, preparation. It goes down to PCR, sequencing, bioinformatics, and so on. We did use existing data standards. So here are two examples. There is a Darwin core developed by Biodiversity Information Standards, or TODWIC, and they are specifically to capture biodiversity data. And then Genomic Standard Consortium developed a MIXS, minimum information about any X sequences. And so they are designed for molecular data. So we took as many fields as possible from those existing standards, but we also created new terms because eDNA is quite unique compared to traditional biodiversity studies or other type of molecular studies that get DNA sequences from tissue samples of known organisms. eDNA is not quite like that, so we created new terms to capture those unique attributes of the eDNA workflows. So those examples are contamination screening procedures or species assignment procedures. And challenge here we had was to find a good balance between keeping as much information as possible versus reducing a requirement for users. So we wanted to make sure that informations shared are complete. And we now have the checklist with the 275 fields, which is a lot, we understand that. And to make it a little bit easier for eDNA researchers, we defined 3 different requirement levels: mandatory, recommended, and optionals. And you can see the mandatory fields is only 36. They are, for example, latitude, longitude, and data providers, primary sequences. So very basic information that everyone has somewhere in their data set. So hopefully everyone agrees that they are all important information, and then not that hard to put them into the standardized data format for those 36 mandatory fields. And another challenge we had was to capture different protocols in one format to achieve machine readabilities, because there are so many different protocols in eDNA studies depending on the labs or regions or individual researchers. And this is where eDNA working group become very active in a discussion, and we've been discussing a lot about what kind of different protocols are out there and can they all fit into our recommended format. And so this is the challenge that we are trying to overcome. And then the final bits of the elements here is the tools and scripts. Here's one example scripts that are the tool that we are developing. It's a metadata validator. So users can format the data first based on according to our guidelines, and then drag that file here, submit, and it produces all sort of warning and error messages. For example, this latitude and longitude is a mandatory field, but it's missing, or this is the controlled vocabulary field, but the entry is not from the list. And so this kind of tool is going to be very helpful for researchers to make the data formatting procedures quicker and more accurate. So what's next in our project? And we are going to publish the guidelines hopefully by the end of this year, and once it's done we want to integrate them into policy. So as I mentioned earlier in the talk, we're working with Wiley journals to work on this, to implement them into the journal publication policy. And then we are working with TODWIC and GSC to register this checklist as an eDNA extension under their standards. And we're going to continue developing tools for data formatting. If any of you have any idea or suggestions of what kind of tools would be useful for you or your lab, please let me know, because those information will be really useful for me to figure out what kind of needs are out there, and we can work together to develop the tools. And we want to run workshops at eDNA labs to increase the awareness about FAIR principles and also about the guidelines, and we can help them to get started with the data set. We will continue revising and updating the guidelines. If you have any, we want to receive as much feedback as possible from the users of the guidelines to continue improving it. And very importantly, we want to harmonize the FAIR and the CARE. CARE stands for Collective Benefit, Authority to Control, Responsibility, and Ethics. And they are developed by the Global Indigenous Data Alliance to protect the Indigenous people's rights and interest in their data. And so FAIR is more data-centric, CARE is people and purpose-oriented, and implementing those two together, CARE and FAIR together, is essential to ensure that the data use is ethical and respectful for Indigenous people and local communities. And finally, I'm really excited that our project is endorsed as an Ocean Decade Initiative under the OBON program. And we hope that we can connect with other projects in many different ways. For example, if there's any project producing a lot of eDNA data, we would definitely like to connect with them. It would be also amazing if we could run a workshop through OBON network. Or if anyone is working in the CARE space, please let me know. I would love to connect with you and then work together to harmonize FAIR and CARE. So this is just to recap the talk. Why do we need to make eDNA fair? Because they are a remarkable and needed resource, but at this moment they're not fair. How are we going to do this? We are working with existing databases, journals, eDNA researchers to standardize the data formats and vocabulary. We're also creating data formatting tools and working to raise awareness. And we will achieve bigger and better data for environmental management. Thank you very much for listening to my talk. I would love to receive any comments and questions if you have any. Thank you.
Miwa, are there any questions from the members of the Zoom call for Miwa? This is great, and I can imagine many groups within OBON wanting to have wanting to have the video that you just showed so that they understand the tools that you're providing, but also have workshops to allow the people that are involved with them to understand how to do a better job of making data fair. So great, thank you very much.
Thank you. Thank you for the comment.
Sophie, you want to go ahead?
Sorry, I think we were muted at this end. I just wanted to ask Miwa, with the workshops that you're planning, will they all be in person or are some of them online? What sort of mode are you planning to do those in?
I'm planning to do both of them. So if I mean, traveling for conferences or meetings and things like that, then I would have to reach out to the labs in that area and then run in-person workshops. We're planning one in one of the eDNA conferences next year in New Zealand. But then we can also definitely do an online version as well, because there's so many tools to do that online, and that's not going to be any issue. So yeah, we're thinking about both.
Okay, well, we'd definitely be interested to have something through OBON as well, maybe at our next OBON annual meeting, which next year is likely to be in Australia, so that could be convenient for you as well.
Yeah, that'd be great.
But we're also planning through our capacity development working group, we're planning some online workshops, and data is definitely something that we're interested in for that as well. There's a couple of questions in the chat. So Becca Millard, are there any tools to facilitate quickly filling the metadata needed? It's so needed, but I wonder how you encourage people to share as much as possible with over 200 fields.
Yeah, I understand. It's a lot of fields, and yeah, we have a R function that anyone can use. We're going to share that when we release the guidelines. So it's basically to with a function you can put arguments like what type of sample you have, how many primary sets you have, what's the name of the primers, and then based on that, it will make a data template, metadata template. So for study metadata and a sample metadata. And then hopefully that would be helpful to have the template at the first, and then you can start filling them.
Okay, thank you. Next question, Stephen Formel. There is no silver bullet, but still have to record these things. You still have to record these things, but groups like NOAA are working on templates and tools to make it easier.
Yeah.
Yes.
So we have a few people from NOAA working with us in our project, and yeah, so they have amazing tool like from what is it called, the eDNA to OBIS, for example. So if there is some, the big database within the lab formatted in the organized way, then we can definitely write a script to format from that format into anything else, and then that could be another way to fill as many information as possible with a script, and yeah, to save some time. Thank you.
Did you want to add anything to that, Stephen?
Yeah, I just wanted to say, so I'm the U.S. node manager for OBIS and GBIF, and I'm part of this group that Miwa is leading. And I just wanted to say that, it can seem overwhelming at first, but what we find when people go to publish their data is that most of this information is already there, but what isn't there is the habits to collect it and, implement it sort of programmatically in your workflow. So I think for a lot of people, it won't be it's not as tedious as you might think. It's just a matter of saying, oh, right, instead of burying the primer sequences in the paper, we will also have them here where we can pull them in as needed, as part of the publishing process. Little things like that go a long ways towards making this not so onerous as it might seem.
Okay, thank you. Just a couple more quick questions before we move to the next presentation. When do you expect these tools to be available? Is one question.
Yeah, so we have a couple of tools in our mind, but most of them will be available by the end of this year, hopefully. And if it's not like a peer review paper style, then we will make it available through preprint as soon as possible so that people can start using it.
Okay, thank you. And how will you make it easy for those without the resources to fill all these fields in for their data? Maybe you've addressed that a little bit already.
So I assume that this resources means like time to fill the data, to fill the fields. Yeah, so some formatting tools would be useful, and then something like what Steve mentioned as well. And yeah, I guess once people are aware about these guidelines and then the format, from the beginning of the study, if they can start using the format and then start filling the data along the way for your own data management purpose, then that could be a way to also speed it up and then not worry too much about the resources and time, hopefully.
Okay, thank you very much. If there's any further questions, I think Frank had a question, but if you don't mind checking that in the chat and then responding in the chat, that would be great.
Yeah, we'll do.
it's more of a comment, I think, than a question. But yeah, if you could please check that and respond, that would be great. So thanks very much, Miwa, and we'll now move on to the presentation by Anaïs on GOT eDNA, guidance on optimal timing for eDNA. So we can see your screen, Anaïs.
Okay, so you see just a slide, which is good. Can you hear me well?
Yes.
Awesome. Okay. Hello, everyone. So today we'll present GOT eDNA. It's a recently endorsed action by Ocean Decades within the OBON program. And GOT eDNA is the acronym for Guidance on Optimal Timing for Environmental DNA. And I will explain how GOT eDNA aims to centralize eDNA observations to guide and optimize eDNA programs. And my name is Anaïs Lacoursière, and I'm a research scientist for the Department of Fisheries and Ocean Canada and the PI of this initiative. Who are we? So we're a group of eDNA experts that accumulated data over the years, over the last decades, working with the statistician, data managers, programmer, and website designer. Some of us are from the Department of Fisheries and Ocean Canada, some are from the Department of Parks Canada, and we have a legal international MOU for data sharing agreement with main eDNA. Being endorsed by OBON is very important to us as we aim to develop the machine learning approach based on combined observation. We are currently developing the algorithms from data collected in Maine, Eastern Canada, and the Canadian Arctic, but with the accuracy of the prediction will increase if we run the algorithms based on larger scale. eDNA research and data increase exponentially, as does the interest in applying it for management decision making. What did we learn over the last decade? We know that eDNA detection rate is extremely variable in space, but also seasonally. For example, we have observed a greater species richness when surveying under the ice in the Arctic. And this is due to the fact that many species have a significant peak of abundance, release eDNA at a specific period. Most species have only a 1 month detection periods greater than 75% detection rate. Better understanding the seasonality of eDNA is important to be able to avoid false negative species detection, reduce the cost of field work and the lab by processing a reduced number of samples. And as any other type of data, we need to know the variation to be able to compare and interpret data between space and time. 2 years ago, I led the International Ocean eDNA Best Practices workshop. It was alarming to see that most of the eDNA groups of experts cannot currently consider sampling periods when designing their program. It means that we're creating a large amount of false negative detection, but also that we're probably use excessive resources than required to detect some species. About 70% of attendees highlighted that this is mainly due to significant knowledge gaps required to adjust their sampling design, and that guidance need to be separated per taxa and region. We just published last year a report compiling eDNA observation from 30 different projects from the Arctic and Eastern Canada. It was the first step of a comprehensive approach to pull previous observation, guide sampling design, and interpret eDNA time series data to guide sampling design. But the report is 300 pages long. If it's only at the genus level, taxonomy level, and ecodistrict spatial scale. We concluded that classical report are not anymore an appropriate communication as technology is evolving quickly, improving your capacity to generate data. And those reports require a major effort and fundings to compile data, analyze the data, interpret results, and at the end, confidence differ between years, region, species, and different groups, scientific groups, altering the confidence from decision makers to use eDNA when taking decision. This is why we believe that the solution is an interactive tool, which we call GOT eDNA. Okay, so I'll start here. I think I'll start automatically. Yeah, okay. So basically, here's the Shiny app. platforms that we have developed. So first we have a pop-up window explaining the limitation of the data interpretation, making sure that people understand the limit of the eDNA on itself. So you can have access to eDNA databases or just use your own data. So if you use your own data, you basically use the tool as a statistical tool. You can use metabolic coding or qPCR. When you use the eDNA databases, that's basically all of the projects that have been compiled together. Then you have in the data request window, you have to select which taxonomy level you're interested on and compile those observations. So you can see all the tags that have been observed so far and compiled within the eDNA databases. So for example, if we select the copy by Lacoursière, then you have all the species that we detected within the databases. You can select all, and then you will have the primers that have been efficient to detect them. They are ranked all the time from the based on the species that you have selected. Then you can see where we have observed that species. So basically you see here the number of samples that have been collected within each region. Here's where I'm based. So obviously we have a lot more samples in our region. So you can see basically the number of samples that I collected in the last couple of years. And then you have basically the observation positive. So we are not reporting the species that have not been detected. And then basically you have, right now we have 4 different models function that is imported within the eDNA. So you can select all of them or just one of them and use it as a reporting tool if you want. How data are combined, we call that the eDNA project ID. So it needs to be the exact same field and lab methods to be combined data. So this is very important to have fair data, complete data sets. Data will not just be imported properly if you don't if you have a missing field from the lab or field. Then you can change the basically the threshold of detection rate. So the default is 75, but if you use lower detection rates, then you have basically a longer detection window. And then you have basically power analysis where you have the saturation curve explaining how many samples you need in a specific month, depending on the species you selected. Everything is interactive, which is great. So Nicole has imported a lot of different functions to zoom in and select data on itself. So you can select it only a month if you want. Everything is interactive. You have 8 maps. You also see the variation within the data for every single month. So you really get more an understanding of the data that has been provided and the power of it, the statistical power of it. And then you have minimal data information that have been selected from the project that has been selected. So. And you can obviously select a lot of different species if you want. So as I said, so let's say you are interested about all topic bugs that we have in the databases, then you can basically see all of them and compare how it differs within a specific group on itself. What I want to show as well is all of the different tabs that we have to help the communication of eDNA results. So every time you're selecting data, then you have minimal informations, and we have data that have been published, some data that are not published, so you can have access to publication. If there is indigenous contribution, then you can see it, and basically if you click on it, then you get access to the information of the indigenous contribution, which is very important for us to basically acknowledge the contribution of each data provider, data owner, and the contribution of the different groups on it. And we're developing interpretation guides and things. So everything will be published, define all of the statistical models for expert and non-experts. So it will be all in GitHub. For example, for the primers, you can see which primer has been efficient and the data you have selected, but also all the full primers that have been used within the eDNA databases so far. If you're interested in a specific question, you can also search on it. So you'll get, for example, which primer have been used for the CO1 or for the lobster, for examples, and which primer are there, and then go back and see which data you need, where it has been detected and has been successful. As I said, the acknowledgement is very important for us because we want everybody sharing and contributing to this initiative. So all of the partners, and we're very happy to have all of them helping us. Here is the working group developing this tool on itself. So if you want to know who we are, it's on the website. And then basically you can always reach out to me if you have any questions in the future. Disclaimer, and an important thing we wanted to add is the glossary, because we realized that people that are using eDNA are not necessarily geneticians. Sometimes they have very low or very high knowledge in genetics. So we want to make sure that everything is defined there. So if you have a questions, you can click on the text, and then you get access to the glossary to better understand what you're saying at the moment. Okay, so there was a platform in itself. When you have a new species in a new location, using the most closely, you can use the most closely related species will be your best bet, basically, to adapt your sampling design. However, some biological factors, such as body type, can be used as a strong predictors, but only for specific months. So the advice, as you can see, the advice is not as easy as most of the eDNA users will like, and that requires some complex analysis and interpretation for the more predictive sampling, best sampling design. So our next step is to have a prediction window that can predict the optimal sampling for a new species of interest or a new location. The confidence of the predictive models is directly dependent on the data that have been imported within the eDNA. As the field evolves quickly, Shiny Apps seems to be so far the best platform, as in science, most of us are using our coding in R, and we just modify and add functions based on our client's needs or lab requirement. And as we improve the eDNA ecosystem models for prediction. Accuracy of guidance is fully data-driven, and thus a strong data sharing strategy is critical. So I'm so happy to see all of the work that Miwa is doing. So as an example, eDNA is possible today only due to the massive effort of sharing sequences for the last decade. So always giving this example, just remember that this is because it has been done that we can do eDNA in 2024. Currently, the accessibility of eDNA data are limited for the development of machine learning. And this is first due, as far as I know, and from the, when people are chatting with me, is the lack of time and resources to format data. So also most data users do not have all this essential information from the field and the lab, so they're not necessarily the expert, but the users. And most lab providers are facing data confidentiality with their clients, the users of the data in itself. And we need an official reporting platform that includes all required methods, information to allow data comparison. If not, we can compare that within, we can compile that observation between space and time. For GutDNA, we did develop a fair data templates for GutDNA that it's pre-filled, so that's saving time for everybody and making sure this is Darwin core, so we can be as fair as possible to save time and ensure strong QA/QC. We strongly believe that having an app where each team from the field, lab, bioinformatics can insistently fill the information will be very, very helpful, and getting all the data together. Too often, data are compiled by the end users years after the project has been done, and the lab and bioinformatics information has already been lost through the various teams involved, limiting us to know which information can be compared. Being part of OBON provides the opportunity for us to optimize our data management strategy and better understand the needs from our various clients. For the functions, for examples, and we are also working with the InBrave groups to link GutDNA to large international databases and working with GBIF as well, as much as possible. Take-home message. So we hope that GutDNA will help to interpret short and long-term eDNA data variation to optimize your eDNA programs. All script will be publicly available in the next few weeks for other scientific groups to use it as a statistical tool if you want. You can use it to determine which methods have worked efficiently for your targeted species from other groups, for example, and guide your sampling design for your program, sampling effort and timing, for example. We made the tool very flexible to add new functions over time, and we are inviting you to build new functions that can be added to the gut DNA app when that will be launched. Our biggest challenge is that the development of machine learning will require sharing eDNA data. So in the long term, we also hope we will be able to secure the resources required to maintain the infrastructure. And a special thanks to the Inuit community who have collected a massive number of samples in the Arctic for more than a decade. A special thanks to all my eDNA colleagues for providing significant resources, exchanging ideas and data, obviously all of the data contributors. And I would like to thank OBON to work all together to implement and improve the power of gut DNA. Here we go. So that's it. My slide, and I'm more than happy to answer your questions, or you can send me emails if you want. Thank you.
Okay, thank you very much, Anaïs. So there's already a question in the chat from Catherine. Is there a manuscript or resources on how the detection probabilities are generated? Are they built only on qPCR data, or do they use metabarcoding data as well?
qPCR and metabarcoding. So we did all of the function. It's all in GitHub. It's not released yet to the public. We have a draft paper, so it's coming very, very soon. We hope before the end of this fiscal year. Basically, this type of tool is infinite. So we wanted to do a very small tool and share it quickly. And then we added functions and tricks and things like that. So basically, we need to stop to polish it and just make it more like an evolutive tool in the future. So we will publish that as it is, what you have seen. And hopefully it will evolve over time with new functions and things. But definitely everything will be open access, and we are very happy if it's if people can debate some functions, so we can develop new functions and things like that. So that's the point, to have it in the Shiny app and all our coding open access. So.
Can I just ask the website that you were showing during your presentation, is that still under development or is that live now?
It's not launched, so it's not publicly available yet. It's on our GitHub, but the GitHub is not open. So basically the strategy that we think is the best is to publish it in a paper with the GitHub. So everybody will have all the details and models and described and things like that. And in the future, hopefully we can have different version, improve and adding functions into it, but it's not publicly available yet, basically. That should be before Christmas, hopefully. Well, at least submitted. Okay, sounds like there's going to be a lot of exciting things happening in the next few months with both of these projects. I don't see any more questions. So thank you again, Anaïs. There might be some questions that come in a bit later, but we'll move on to Katie Pitz's presentation on BeBOP.
So, thanks, Sophie. I'll start sharing my screen. Let's see if that. Okay, great. Can everyone see that okay?
Yes, perfect.
Thanks. So hi, everyone. My name is Katie Pitz, and today I'd like to share with you some work we've done as part of the Better Biomolecular Ocean Practices Group, or BeBOP. And I'm presenting work that was done on part of a large group of people. But we came together in 2020 with the question of how can we better share, discover, and compare omic biodiversity protocols. More and more omic protocols are being used to generate biodiversity data, but often these protocols can be changing very fast and are quickly outdated or quickly improved upon. They can be hidden in papers and hard to implement practically, or perhaps the lab has moved on and has now changed what protocol they're using. And furthermore, understanding the details of these protocols is often critical to understanding the final biodiversity data products. So as a group, we thought we needed the ability to directly compare protocols between different lab groups. We wanted the ability to discover what protocols may be best suited for whatever your research goals are or the region that you're working in. And we also wanted the ability to be able to track protocol changes through time and also link unique versions of protocols to their resulting data sets. And so together we came up with a couple different solutions. And here's one figure from our paper in 2021. But our first solution was thinking about, instead of a long detailed protocol that was all one document that combined many different steps together, we thought instead you should break out your protocols in these different modular units, like having a protocol separately for field sampling, for DNA extraction, for PCR amplification. And then we envisioned kind of a chain of protocols occurring. So all the different steps that occurred that resulted in your data set. And these all could be linked to your unique data set. And so we designed protocol templates in the Markdown language. And the advantage of using these templates is that they're more machine readable than a Word document or a PDF, and that we're able to track changes in our protocols through time using tools like GitHub. And then also by developing templates, it makes it easier to compare protocols for the same step between different labs. And we also wanted to allow protocols to be more findable and searchable. And so we also wanted to develop terms to describe the protocols. And so here I'm just showing different large category bins that we created. We call them methodology categories, but they're just bins to place these protocols in that helps organize them. So if you wanted a field sampling protocol, you could look inside the bin for sampling collection procedure. But together we kind of developed a set of metadata to describe protocols called minimum information about an omics protocol, or MIOP. And this metadata is what will allow you to find and sort and determine which protocol would be best for your use case. And so the first term is that methodology category, that large bin for what type of protocol it is. And then we also developed terms that describe what are the goals of this protocol. So like, what project was it developed for? What is its purpose? What analyses does it contain? And what's its target? We also use different tools to describe the environmental context that that protocol was designed to work in. What is its location, its broad scale and local environmental context, and what environmental medium was it meant to work with? And then we also have terms that describe what are different requirements for that protocol. So different materials, skills, time, or people that may be required. And then finally, we have a set of terms that helps you kind of sort and catalog the protocol. So what language is it in, who was the creator, and what's its maturity level. So together we thought these terms describing protocols would allow you to search for the right protocol for you, depending on what your goal of your project was, maybe where you wanted to work, or say you were doing field research in a location that limited what materials were available to you. You could see and limit what protocols you wanted based on that information as well. So together we've been working to develop these tools on GitHub, and we've created a GitHub site that houses our protocol templates as well as our metadata standard, the MIOP terms, and we've added some guidance on how to get started as well with sharing your protocols using our templates. But I thought I'd kind of first walk through what it looks like practically. So here's one of our repositories that contains our protocol templates. And so far we focused mostly on the laboratory analysis. But each of these markdown files starts off in a similar way with a table for the metadata about that protocol. And then it has several different sections, including a section for the standard operating procedure, the actual kind of protocol of what you're doing in the lab. And so this is just highlighting how in the template, we try to pull out information that we consider important for that procedure. So here's from our PCR template, where we ask you to pull out the primer name and sequences, your reaction mixture, and your cycling program in a standardized way. So I thought I'd highlight a little bit about what this looks like in practice, because there's been some hard work done by the Ocean Molecular Ecology Lab at NOAA led by Zach Gold, where they've taken some of our templates and shared their protocols on GitHub. And so here on the left is just a image of what their site looks like. But then if you scroll down, they also have links to their different protocols that they've shared using our template. And you can click on any of these links, and then it brings you to their protocol, which always starts off with those MIOP terms. So here you can see like where the geographic location this protocol for 16S PCR they've applied in. So different terms that kind of describe this protocol. And then again, like going down to like the PCR section, they list their primer sequences and these different tables that we ask people to include, making it kind of easy to immediately see that important information. I also want to highlight another lab at AOML with work by Luke Thompson. But here you can see they've also started sharing their protocols in this way, and zooming in on one of their sampling protocols. Again, it's that MyOp table. And then I just wanted to highlight too, we ask people to include important equipment that is in use for the protocol, so that depending on your sampling situation, you could decide if this protocol would work for you or not. And so the actual steps of the protocol, and then finally at the end of our templates, we have a section where you can talk about different quality control measures or basic troubleshooting, and link references or data sheets that you would like to share with others about this protocol. So far I've shown you what we've done in GitHub to be able to use these templates, but we also understand that GitHub may be not quite as user-friendly for those who aren't used to using it. And another tool that has been used a lot in the biomolecular community is protocols.io. So similar to GitHub, we put our exact same templates onto protocols.io. So if you're already using this tool, in the same way, you can copy our protocol templates and then fill them in with your protocols. So here's an example of the template on protocols.io. And then here's an example of one I filled out for our lab for our 12S PCR protocol. But similar to GitHub, you start off with that MyOp table and then walk through the different steps. And so one of the nice things about both GitHub and protocols.io is you can get DOIs for unique protocols. You can track changes in these protocols. And by using these templates, your protocols are a lot more easily comparable between groups. So here's that PCR section again. But one further unique feature or nice feature of protocols.io is you can create chains of protocols together and then get a DOI for that unique chain. So here I've used our templates to link multiple steps together. And then you can get a DOI for this unique linkage that you can link then to your data set. So going back to our original vision, that for every data set, you'd have like a string of protocols that is associated with that data set. And we would like to think that if you needed a protocol in that chain, like a field sampling protocol, you could then use these different MyOp terms to sort and decide which field sampling protocol would be best for your situation. And so far as this group, we've focused really on laboratory protocols to get started, but we'd also be very excited to think more about how we can develop templates and encourage submission of protocols to deal with the other parts of this workflow. So here, this is the typical meta barcoding workflow, but we'd like to also have protocols that capture how the experimental design happened, how you got permits or permission to work in the location, your data management plan, maybe how your samples are being curated or stored for further data analysis down the line, and then how you're releasing your data and publishing your results. And so both these areas are also very important and often not captured. And so as part of BeBOP, our continued activities are continuing to collect and map protocols and data workflows. MyOp has now become a project under the Genomic Standards Consortium as well. So we hope to transition that metadata standard to be hosted there as well. And then the work with the protocol templates, we continue to improve and make more protocol templates. But we'd also like to do kind of a comparison between common protocols in use between OBON projects, like focusing on PCR application, for example, to see what the landscape of PCR primers being used is between projects. So I'd like to thank everyone in the group and also the work on behalf of Zach Gold and Luke Thompson to start using these templates. But thank you very much.
Thank you very much, Katie. Are there any questions, Margaret?
Not from me. but I hope that we can have a little discussion amongst all three of the programs in terms of linkages and interactions between them.
Yeah, I on that note, I was wondering with in Miwa's presentation, I got the impression that there was also an objective to share protocols, and I was wondering how that might interface with BeBOP. Miwa, do you want to clarify that or comment?
Yeah. So I was can you hear me?
Yes.
Yeah, okay, good. Yeah, I really enjoyed talking to Katie, and yeah, I think we should definitely talk about our works together because I think there are some overlaps, and then we can definitely yeah, work together to do it. But basically, yeah, in my work, we have a checklist that also describes the protocols, so sampling methods and then how the PCR was done, extractions and so on. And we list up the name of the field names in a way that we can capture those protocols in a machine readable way. So you might be doing something similar, then we should I'm really happy to show my checklist, and then you can identify what is overlapping. And then some of the names that you showed in your slides, I recognize them and I'm using the same one. It's probably because we source from the same standards, and which is great. So yeah, so we are also working on the protocols as well as the output of the data, like the results of the eDNA studies.
Yeah, that would be great to connect more. We also try to reuse metadata terms whenever possible and not just create our own or new ones.
Yeah, we're the same, so we're on the same page.
Well, as I mentioned earlier, the plan is to have a protocols working group within OBON. Until a bit earlier this year, we had BeBOP as an endorsed project. So that was kind of our go-to for protocols sharing, discussions about protocol sharing. But that's come to an end as a project. So we're sort of transitioning to having a standing working group. So we would very much invite all the OBON projects, and particularly the ones that are in this webinar to participate in that working group. So yeah, we definitely need to try to converge rather than having too many different or slightly different versions of things, so that we can kind of encourage the community to go to one or a few places rather than having lots of different options. Okay.
Stephen Formal has a question. Are there any people who are in both GSC working groups?
And that I'm not sure. I think Nick Jeffrey was he in there? Maybe he was in Anaïs project actually. There might be.
Yeah, I think there might be. I saw Luke's name from NOAA in your slide, and
yeah, he's been starting to use the BeBOP
with that. Yeah.
Yeah.
Okay, maybe we can also compare those collaborators, and then we probably have some people working for both of us.
Yes, and Luke is actually participating today. I don't know if you want to
Luke is here. Okay.
He is.
Yeah.
Hey, hi guys. I'm dealing with a cold, so I'm staying quiet, but yeah, happy to help make those connections across the different groups, make sure the terms align, and thinking about where in the sort of process we can integrate these different reporting methods. So yeah, it's really exciting to see it all coming together and getting close to being where everyone's using these routinely. So thanks for all the efforts.
other comments or discussion?
Is there another comment in the chat? Sorry, I'm just struggling. Frank, maybe Frank, do you just want to make a comment verbally?
Well, there's a lot of other groups that are not necessarily part of the Ocean Decade that are important to network as well. And so I wonder how we do that. Luke, who just spoke, is also basically leading an effort in the Marine Biodiversity Observation Network to try to work on some of these ideas and basically to network people around protocol standards. Katie's been very active. Many of you have been involved. And as we think about how the decade can be inclusive in a way, if people don't write a project proposal, then you're not endorsed by the decade. And yet there's a lot of people that we want to network. And you're doing that anyway in the way that POGO operates. But I didn't know that BeBOP was ending. And so that I think that that's fantastic that you're picking it up as a project to continue it. And so that, but you have the right people here, so I don't have much to say. I'm really happy to see that. And I just hope that we can continue to see that openness in the networking that we do.
Thanks, Frank. Stephen?
I think Anaïs might have had her hand up first.
You're in the reverse order on my screen, but ladies first.
Okay.
and Anaïs,
yeah, for me, it's a quick comment, so I'll jump in and then Stephen can jump in. So first, thank you very much for organizing all of that. And you know, we are most of us part of like different groups and things like that, so we see that evolving. And obviously, the templates we're using is match well with Miwa templates that we've communicated a couple of years ago, and I'm working with Katie on that. So we are, this evolving. What I found very hard is to change practices from the labs and the different groups because we're accumulating data. So we're trying our best for providing mapping as much as possible. So at least if someone is using a different wording that are similar, they can do that. Some things that we're doing with Brave and GBIF. And yeah, so thank you very much for giving that opportunity. I think we're getting there slowly, but we're getting there.
Yes, and the next OBON meeting will be an opportunity for many of the new OBON projects to meet one another, and so I think that will be a really good opportunity to continue some of these discussions.
Yeah, definitely.
Thank you, Stephen.
Yeah, so I think one thing that I wanted to point out is that it's good that where these groups are connecting. Because an important part of Miwa's work is that TADWIG aspect, which is about connecting eDNA to the larger biodiversity world and making sure that eDNA data can work with other types of biological observation or biological evidence data. And I think if you're very focused on eDNA or other types of, very specific studies, that might not be obvious to you that there's use for your data well beyond its primary purpose and even beyond the eDNA community. But then I also had a question for Katie, because one of the things we've noticed in trying to implement any types of biostandards is that having examples of using it is one of the best ways to get other people to use it because they feel comfortable, they recognize their own work in it. So I'm curious if you have an example of the BeBOP protocols being cited in a paper that we could see, so people could go to that and say, aha, this is how BeBOP ends up in a publication.
Yeah, I'm working on a paper right now where I put my protocols into the BeBOP format on protocols.io. So I'm hoping that will act as also an example. We did an initial paper in 2021 showing kind of our idea and the goal of the project, led by Rob and Samuel. But yes, we need to do more examples of using these protocols in a published science paper. and how that makes it easy to reproduce the workflows. You're right. I think also an important component though was like of this project is trying to figure out how can we better share what protocols are in use right now, so that you don't have to look at a paper and maybe know what people were doing a couple years ago when they collected the data, but also what are groups doing right now when they're currently collecting samples or analyzing samples.
That's a really good point. Thank you.
How can we, well, let me back up. Early on, OBON said, we're not going to get into the business of trying to tell people how to work with their samples, i.e. that we weren't going to say, oh, here's the gold standard and everybody should do this, but rather focus on making sure that people included all of their protocol information and shared that and their methodology so that you could understand what the differences might be in your data and other people's. Those of you who are working in this all the time, how can we foster that conversation and kind of help this whole process evolve? What can OBON do to help?
I think I've seen a lot of wish for guidance on knowing what protocol should I use for this situation, kind of, or like, you know, or there's situations where everyone's trying to figure it out together and they'd like to know what other people have tried so you don't waste effort trying the same thing somebody else has done. So I think maybe if the wish and need for that kind of guidance might drive a lot of people to share what protocols they're using and what has worked and not under different situations. So it might be that OBON could kind of host either workshops or discussions where people have shared kind of things that haven't worked for them or things that have, and then in that way kind of ask for people to share their protocols, so we can know what people are doing and where.
Okay.
And yeah, I have something else to add. So it's a really good point. And also I think, you know, my project is about data formatting. So we want to standardize the format of the data to be shared, but not about standardizing a method of the process before the data is generated. So when I introduce about a project, I didn't do that today, but I should be highlighting that, you know, it's not about standardizing the sampling method or extraction method. It's not that part, but more about sharing what methods were used in the standardized way. So maybe when you, as you know, OBON programs, when you know, we have multiple projects like this and Katie's, and then when you introduce about those projects, you might want to highlight that at the beginning, and then that way you can still align that with your goal.
And maybe I can jump in there. And I think it's a very important discussion actually, because I'm completely not supporting standard right now for like the same standard for every species or the same for all type of ecosystem, but at least I think we're moving towards seeing, having indicators of, that's what we're trying, got eDNA to inform which method have been more efficient in that specific location and things. And if we link our platforms together, Miwa, Katie, like all of those methods, over time, we will see which one has been the most efficient in that those locations and things like that. So the depth is a good example. Primers are, you know, there will never be a perfect primer, and then technology will evolve anyway. But if we are able to have platforms that communicate well together, this is actually a very, very big step, because right now what have been done in the last decade is publication, and it's impossible for us to compare one publication to the other, the results on itself. So if we are getting, I think we will get there by just ensuring that all of our platform are linked, so databases all together with data analysis and protocols basically. So. And sequencing and bioinformatics.
I think if Pierluigi were here, he would say that the system to link all these systems together is ODIS, the Ocean Data and Information System. I don't know how, if you're all familiar with this. It's being developed by under the leadership of IODE, and it's the vision is that we don't need to make things interoperable with each other as long as they can be harvested by ODIS, which can just kind of gather. As long as the information is there in a structured format, it can be harvested by ODIS and discovered kind of through a central portal. So that might be worth some further discussion about later on, and to see if I know we've had discussions already with BeBOP about this, but maybe this is new to the other projects. Sorry, I jumped in, but there was a hand. I think Catherine had her hand up.
Yeah, I was just typing my thoughts, but you know, I think Katie mentioned that folks are often looking for guidance, and even if someone's not giving direct guidance, like this protocol is best, at the very least, like having a way to search BeBOP protocols and, you know, searching by those metadata fields that Katie was talking about. So I don't know if OBON or someone else could host a website that, you know, is a little more user-friendly, a little more searchable to collate like BeBOP formatted protocols. So basically like protocols.io, you know, provides a platform for you to search for protocols. Can we do something like that with these BeBOP protocols that we're putting together?
And if I could just jump in too, I was really excited to see on Anaïs got eDNA platform that there would be a way to see by primer set which ones were better at detecting different taxa. And so I think that's a really powerful tool too. So you could see using these CO1 primers versus those CO1 primers where you've detected that taxa in that particular environment. And so I think linking these things together of like, okay, using this protocol, this primer set, this is where you see your taxa of interest would be really great.
And we have worked hard for basically very complex algorithms and stats for, at the end of the day, it looks very simple because we realize that people that doing eDNA have very different expertise originally. So the communication has been already very complex. So, and making it more flexible at the end, it's like as a Shiny app, it's great because it's evolving quickly. So it's really giving the opportunity for us to improve that type and having those discussions with all of the groups and people trying it and getting their feedbacks. Because if you, let's say someone is giving a try and get the DNA and does not like it and then is not using it, it's not really the point. The point is to send us like why for your program it didn't work well, so then we can adapting it, and why you cannot combine with data, or why you don't want to share your data. So give us the opportunity to have a file that is maybe just a project that is not shared. You know, it's just observation, and you don't mind to share the special example trends, but you cannot share the full protocols yet. So giving that feasibility is now easy with the technology we have, but we need to really get a feeling on why. What's your limitation based on your specific role? Species at risk don't have the same concern than species invasive species, for example. So we need to make sure that is well communicated to us and working all together.
Are there any more questions or comments?
I'll just make a comment. The Capacity Development Working Group of OBON has been struggling with, you know, what are the tools that we can use to help people get into this? And, you know, I think coming into OBON, we thought, oh, you know, it's going to be a lot of workshops. You know, we're going to get people to do workshops for those that are new to eDNA or new to a particular type of analysis. And what we've been learning is that aside from the cost of doing workshops, I mean, we were talking about physical workshops and getting people there and so forth, that just isn't reaching a lot of people. And we've been talking about instead trying to harvest all of the online training and presentations and whatnot that are available and group them into sort of sections like sampling, method, analytical methodology, data management, etc. And then within those, try to class them as, here's something that will be really useful for somebody who, a graduate student who's never done this before, versus here's something that would be useful for an expert who is working in a new area or doing something new and needs a high-level discussion. And as I listen to all of this, I'm thinking that just the presentations that you had today would be a really great addition to that whole constellation of things. But especially those of you that are thinking about methodology and training and making more things available, keep it in mind. And I'll certainly reach out to the 3 presentations, but if any of the rest of you are thinking about this, we would love to incorporate you into the capacity development working group as we talk about these items. Where Sophie's putting out a call to everybody associated with OBON to make us aware of any online training or presentations or PowerPoints, whatever that you're aware of, so we can start doing that. So we envision a resource on the OBON website that would allow somebody new to come in and get started and then be able to go back and go to the next level or whatnot. So any comments that you have on that, or even on the wisdom of trying to do it that way, would be helpful.
Yeah, so running workshop is really our next milestone once the checklist is developed. And then, because I think even if there's like a written document on the website or manuscript, it's hard for eDNA researchers to read through and then actually start using. Whereas if there is like a few-hour workshop that we can actually get, hands-on workshop with their own data set, we can help them to get started, then that's going to be a really effective way for them to start using it. So yeah, if there's any opportunity to run a workshop and if any resources are limiting factor, then online workshop is going to be much cheaper to run, I think. And then my time is there already. So yeah, I will be happy to contribute. And if it aligns with the capacity building objective of OBON, then I would be happy to join that team as well.
Great.
And I would like to add then after a workshop, I found very helpful when everybody write a paper together. It can be a very short paper, but at least showing how to link our different project is very initiative, or where's the field and what's the gap. So it really helped people to word what's the limitation basically. Often we know we can't, but we don't really take the time to really think about how it can be possible politically, technologically, and things like that. So wrapping up a manuscript after workshop. I think they are the best papers, even if they're very general and there's not new science. It still make people thinking about how to move forward. So personally, I like those papers.
Great. Catherine?
Yeah, I think your comment about a web resource is a really great idea, because that's something that people all over, they're searching for like ways to do eDNA, can find very quickly. And yeah, I think when you think about like accessibility to these workshops, like we often know the people who go to them, and it's harder to reach people that you might not know. One idea, or just something to think about with these web resources, is to make sure that it's has a really good user interface. It's very searchable. It's very like, it has the terms that will make it pop up on Google, you know, like have web developers and folks involved in putting that together. So it is really accessible to people who are new to the field, as well as people who are maybe experts coming to see what else is going on.
Yeah, great point. Thank you, Catherine.
Yeah, we had a bit of a discussion about this in the capacity development working group, but kind of been wondering how, again, not trying to create too many different places for people to look for protocols. So we're talking about having, you know, potentially a resource that's aimed more at people who are getting started with eDNA, with maybe protocols that have videos associated with them. And then there's BeBOP, which is kind of more technical. It's in GitHub, which can be a bit of a barrier. I'm kind of wondering if there's a way that we can link these two, so that we're not kind of, again, doing things in silos. I don't know, Katie, do you see it? Do you see it maybe a future development for BeBOP where protocols could be exported in a more user-friendly format, or I don't know, videos could be added or something like that?
Yeah, so I mean, with the protocol templates as they are, you can export a PDF of what, you know, or a document of what they are. You don't have to have the markdown feature. And then I think like what Catherine brought up would be a great feature too, is like that we want to have, is using those MyApp fields, be able to select for a certain protocol based on where you want to sample or what you want to do. And so I think thinking about how to link things would be great. I know one idea that's always thrown out is like a decision tree, or like something that aids a user in deciding exactly what protocol to use. And so thinking about a smart way of designing that based on all the resources that we have available to us, I think would be really great.
Okay, well, I think we've reached the end. Frank, you have a quick comment?
Yeah, sorry for the late question, but you're organizing a meeting at the Conference of the Parties for the Convention on Biological Diversity in Cali coming up soon, Sophie. And I was wondering who here was going. Maybe, I don't know if Margaret, if you're going.
I'm not.
But I wonder to what extent that is an opportunity. I assume that you're going to talk about OBON and promoting these methods. And yet, when I look at the Convention on Biological Diversity, they have this coming Montreal Global Biodiversity Framework. There's only 2 indicators that they have included in all the targets that have anything to do with genetics. And one of them is this NE 500 indicator, which some people say doesn't work well for the ocean. The other indicator is not really even about the genetics, it's just how many species are being tracked with DNA methods. And so it's kind of more of a tracking management more than tracking biodiversity. And so I wonder, where do you see this going? Because there's so much, and maybe it's because it took so long for the CBD to get these indicators, and most of them are pretty difficult to understand. They don't really fit the targets well. They don't fit the goals very well. And so that whole process is a bit not as well organized as one would like to see. But what are your thoughts in terms of indicators or the application of these methods to these international frameworks, and especially how different countries are going to use them to report to the CBD.
Thank you. Difficult question. I don't know if
No, and I'm not expecting a clear answer either. Maybe I was looking for what are the opportunities and how, because I think this is the group, and many people here are in the different groups that are doing this for the ocean, and yet there's no real good indicator yet included that uses genetics in any of these frameworks.
Yeah, well, it will be the first time for Pogo being present at CBD COP, so I think it'll be interesting to engage and find out a bit more about the targets and the indicators and how how all that's going to work. But we have, as I mentioned, we're looking at setting up an OBON policy working group. We also have one of our future webinars will be with projects that are kind of more application oriented, so fisheries, MPAs and things like that. So yeah, I think this is something that we'll be looking at more closely, how to engage with the CBD process, among others. So watch this space. Okay, well, we're a little bit over time, so I'd just like to thank all the speakers once again for all the great presentations, and I think this is the beginning of a lot of new collaboration, which is great. And Luz has posted some links. If you missed the last webinar but were interested, the recording's now available on the OBON YouTube channel, and there's a link there, as well as the link to register for the next webinars. And the next one will be on Monday at 4 o'clock UK time. So thanks very much, everyone, and hopefully see many of you on Monday.
Thank you.
Thank you very much.
Thanks, everyone.